Okay, today we're going to talk about the amino acids. A couple of Poggle questions that I want you to go ahead and get on here is I would like to see you draw the Hayworth projection of D fructose. So I'd like to see that labeled and drawn. So I want to see the Fisher projection, and then I want to see its Hayworth projection. And I want to see you do that, draw that, and show that Mutter rotation, where it can go from the acyclic to the cyclic. That's one thing I want you to do. Number two, I want you to draw structures that show the difference between the structures of DNA and RNA. Okay, so I want you to tell me the difference between the DNA and RNA, and I want you to include some structures in that. Okay, and then the third thing that I want you to do is I want you to draw out your amino acids structures, and I want you to classify them. Classify them into the 4 different categories that we're going to talk about here. So basically today's in this video instruction, you'll want to take notes and submit those because I want to see the amino acid structures drawn out. And so how many amino acids are there? And you do have to know these. There are, let's say 20 amino acids. So you need to have, AA is what I write for amino acids. I want those drawn out and classified. We're going to do 4 different categories. So amino acids are your proteins. So amino acid is the monomer, and the polymer is called a peptide. And so your amino acids, and when they're in order, so when they're linked to make the polymer, this is called your primary structure of protein. And you're going to learn and classify the different structures of protein. There's primary, there's secondary, there's tertiary, and quaternary. And I need you to be able to classify all 4 of those. Okay, so your structure of proteins, you have a primary amine, you have an alpha carbon, and then you have a carboxylic acid. And then you have different groups that come off as the alpha. So these are called alpha amino acids. And you have different R groups, you put R, different R groups that come off the amino acid to make it unique. And I want you to draw each one of these out. So each structure, I want to see NH2, and then a carbon, and then your carboxylic acid. And then I want you to draw your different R group. So if you have a CH3 here, this is called alanine, okay? And so you can divide your paper up into 4 sections. And this one's going to be hydrophobic, and you can have hydrophilic, okay? This is also hydrophilic is water loving, so that's apolar. Hydrophobic is nonpolar. And what determines this is these side chains here, okay? So it's these R groups that determine what 4 blocks you'd put this in. And then you're also going to have basic, based on the R group, and then you're also going to have acidic. And you want to go through each one of these structures and draw them into one of those 4. Okay, so we have alanine. Serine would be your hydrophilic. So serine, you would want to draw NH2, CH, COOH, and then serine would be CH2SO. Or sorry, not SO. OH. Okay, so it is a primary alcohol. Okay, you see? So that's a primary alcohol. And you're also going to see there's three alcohols. We also have um, we have threonine. Okay, so that's an alcohol here. And you'll see that is a secondary alcohol. And then the other alcohol we have, and these are all going to be polar, would be a phenol. And that would look like this. OH, and this is called tyrosine. So you need to classify each of the amino acids. We got alanine. If it just has an H, if R is an H, that's glycine. Okay, and that's the only one that is achiral. Every one of these are chiral, and they are L amino acids. And when you put these together to make a primary polymer, so primary structure, you go from the N and you do NCC, 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 and this is going to be a tripeptide. Okay, so if I want you to give me, and you might get something like this on your quizzes, I want you to pick out the structure for, let's say, and you have all those there. Let's do, sorry, let's do alanine. Let's do ASN, that's asparagine, and let's do CYS, which is cysteine. Sometimes you can also have these as three-letter codes, so that would be alanine, asparagine would be N, and glutamine would be Q, or not Q, we don't want glutamine. Cysteine is C. Okay, so then sometimes, so you'll either get them listed as A and C, which they don't actually even have a line through them. So you would just get like A and C, or you could get dashes like this with the three-letter code. So if you were to draw that primary structure from given these amino acids, you would do an H2, this is your alpha. Okay, what's alanine? Alanine is a CH3 here. This is your peptide bond, okay? So it is an amide right here. So you have one peptide bond here. And then you go to your next. This is your alpha hydrogen there, and we need asparagine, which is CH2, and then this is asparagine. Oh, yeah. Okay, sorry. This is an amide. Okay. So you see that's an amide. What would an amide be? That would be hydrophilic. Okay, so amides are hydrophilic. It means they're water soluble. All right, and then the CH3 would be hydrophobic. And then we have another one, which would be NH, and then your alpha hydrogen and your carboxylic acid here. And this is lysine. Lysine is got 4 carbons. I'm just wondering where it is on your slides here. Polar, you got your polar, you got your acidic, it would be under your basic, and you have CH2, 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 NH2, okay, which a lot of times would be protonated at physiological pH, but there's you have your primary amine. This is basic, okay, so this is a basic, this is hydrophilic, that's hydrophobic, and that's how you classify these in your division here. You should have 3 basics, amino acids, and you should have 2 acidic amino acids. Okay, so the first slides you have, when you go through the slides, are all your different amino acid structures. I would recommend that you make index cards and you learn these and you classify them. And so just other things to know besides classifying your amino acids and knowing their structures is that they have different pKa values. Okay, they have high melting points. They're more soluble in water than ether. The pKa value of this carboxylic acid is a pKa of 5, so that doesn't surprise you. But the amine here is got a pKb value of 4. So we're going to talk about the different pKa values and how that determines its isoelectric point. That's coming up, okay? But before we get there, let's learn another word, okay? And it's called a zwitterion. Zwitterion. A zwitterion means there's a plus and a minus in the same molecule. Now let me just tell you, that makes that very hard to isolate and work with in the lab. So the thing is, with your amino acids, you have an amino group and you have a carboxylic acid group. And so this can get protonated and deprotonated. So if you protonate the nitrogen, it becomes a positive charge, and you deprotonate the carboxylic acid, it becomes a negative charge. And this is a overall neutral, but it's called a zwitterion. And so the actual structure depends on the pH of the solution. And a lot of times the way scientists will isolate different proteins is by changing the pH of the solution. So different R groups, it's all about the R groups, get protonated. And that would be basic. Remember, there's 3 basic R groups, and different R groups can get a negative charge, and those are your acidic, okay? And those are 2. So these are the only 5 of the amino acids can actually get protonated or deprotonated in order to purify them. Okay, so one of your slides, that's what it's talking about, structure and pH. Now when you're just drawing them, you can just draw them as the neutral. So I would just draw them as the neutral, and then you just focus on the R group. Okay, you don't have to worry about that. But when you have a solution and you're changing the pH, so you go from 1, 2, 3, 4, 5, 6, let's say we go all the way up to 12. Okay, so let's make these more of increments of 10. 10, 8, 6, 4, 2, and 0. Okay, now, so you can see, and draw that here, and if we're dealing with a structure such as just, I guess, without any R group, so this could just be glycine. Okay, so we look at glycine, and at low pKa, so this is a pKa of 2.3, okay, we're adding base. That's how the pH starts low, and then it's going to go to basic. 12 is a basic pH, okay, so this is basic. Lots of sodium hydroxide, okay, has been added. So what happens, what does your structure look like of glycine at pH of 2? So a pH of 2. Well, the carboxylic acid is protonated, and so is the amine. Okay, everything gets protonated. And what I forgot in here was my alpha hydrogen. Okay, so everything's protonated at low pH. You've got lots of protons. Lots of protons. Acidic pH, you got lots of proton. The nitrogen gets protonated, the OH gets protonated. So what would your basic one look like glycine? That would get protonated, and then you have CH2, 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 NH3. Okay, the side chain would also be protonated. And then you get here, this is called the isoelectric point. And that's where your molecule is neutral. Okay, so what is our molecule right now at pK of 2.3? It's overall a positive 1 charge. Isoelectric would mean, let's put in our isoelectric would mean that this is minus and this is plus. Okay, so minus and plus, that's neutral. So the zwitterion would be, this is where your zwitterion is, the isoelectric point, and that's at pH of 6. And then you go up here to pK of 9.6. Okay, we're under basic now. So your carboxylic acid will be deprotonated, and everything's deprotonated. So NH2. So now this is overall a negative 1 charge. Okay, because everything, no protons in base. So the isoelectric point of amino acid defined as the pH at which the amino acid exists at the zwitterion. Okay, so amino acids, the acidic, so when you have your amino acids, this portion here, the isoelectric point is about a pH about 3. Okay, that's when it's going to cross over to the negative. And then the amine, the isoelectric point is about 9, pH about 9. Neutral amino acids, pH is slightly higher, about 5 or 6. Okay, so actually, it's not really about this. Okay, so let me just tell you what this is again. Okay, let's do this. This slide's kind of important here. Okay, so you do need to write this down. The acidic amino acid isoelectric pH is around 3. The basic amino acid isoelectric point is around 9. And then your neutral amino acids is around 5 or 6. Okay, so let's not even worry about that so much. Well, okay, we'll put it in there. Neutral, 5 or 6. So if you remember, we have about 3 basics. So let's just talk about lysine. That means this is going to be at pH, so at pH 9, this would be charged, not charged. So anything lower is going to be, so if you do a pH around 6, this is going to be an overall +1, because these are going to cancel each other out, but then you're going to have this. The reason why this is important is if you read about electrophoresis, separation, okay, you have this separates by isoelectric points to separate the mixtures of the amino acids. So if you have a positively overall positive charge, it's going to go to the cathode. And if you have a negative charge, it's going to go, the positive charge are attracted to the cathode. Okay. The negative charge would be the anode. Wait a minute. Okay, so sorry about that. You know what? It's really hard to actually make a lot of corrections. So we're just going to start all over. Like I can't actually do the insertion. So let's just get through this. So we're talking about electrophoresis separation. And so when you have a compound that is overall positively charged, okay, then it's going to be attracted to the cathode. And the cathode happens to be negatively charged, okay, because opposites attract. So this is your amino acid, and if it's a positively overall charged, you're going to find it at the cathode. Whereas the anode is a positive charge, and so it's going to attract the negative overall charged amino acid. An amino acid is isoelectric point. Is it going to move? No, it's not going to be attracted to the anode or the cathode. So you have a nice slide that shows how to put a certain pH buffer, and you have an anode and cathode, and then you start the charge, and you'll see that your basic amino acids are positive. So all your basic amino acids like glycine, overall charge, the R groups, are going to be attracted. This is a pH buffer 6. Are going to be attracted to the cathode. Okay, so it's going to go towards the cathode. And then your amino acids that are acidic, they'll have a negative charge, and they'll be attracted to the anode. And this is how we can separate the different amino acids. The other thing I need you to understand is the difference between primary structures, secondary, tertiary, and quaternary. So please, in your reading, be sure to look at that. We've talked about primary structures so far. We did like alanine, maybe glycine, and cysteine. So that was primary structure. You have for secondary structure, it's the hydrogen bonding between the carbonyl oxygen and the NH bond of the amide. And you're going to get two kinds of structures. You get a thing called the alpha helix, and then you also get a thing called beta sheets. Okay, so look in your slides and know that, and look at those structures and know that it's the hydrogen bonding that is the secondary structure. Tertiary structure is the most interesting, I think. Okay, and that's your R group. So this is why you have to determine whether something is acidic or basic, your R groups, hydrophobic versus hydrophilic, okay? And the types of tertiary structure you have is hydrogen bond. This is hydrogen bond between R groups. You have also sulfur, disulfur. It's called disulfide bridges linkage. Okay, and that's a sulfur-sulfur bond. You also have van der Waals, which is hydrophobic. Okay, same thing, hydrophobic. And then you can have electrostatic. And the only ones that can do this would be your basic R group, that would end up being a positive charge. And it would do an electrostatic with the negative amino acid R group. Okay. And then you also have 5 hydrophilic. And so you can look at those. The tertiary structure determines the folding of your protein, and the folding determines your function. Insulin is really interesting. It only has 21 amino acid residues, and it forms two disulfide R group linkages. So the tertiary structure is known. You see your slide, it's slide 36. A lot of people that are born type 1 diabetes, sometimes their insulin does not have the disulfide linkage because they're missing an amino acid. You do have a lot of slides talking about protecting group and synthesis. We're not doing that, but you do need to look at your slides covering the different levels of protein structure, primary, secondary, tertiary. You have slides showing the alpha helix and the beta sheets and how the hydrogen bonds, so you need to be able to label that. Tertiary will show you the different R groups. Quaternary structures, more than one amino acid. So when you have NCC, 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 excuse me, that would be one amino acid or one peptide protein. And then you could have another one that starts with NCC, NCC, NCC. Now keep in mind these would be big. And then if those two come together, then that would be a quaternary structure. Hemoglobin. Hemoglobin is responsible to carry your oxygen through your blood to your different cells, and it has two alpha units and two beta units. So it has four polypeptide structures that come together to make a functional hemoglobin, and that would be quaternary structure. The other word I need you to know is denaturation. So denaturation, and this is what we have to worry about with different pHs, temperatures, even mechanical, and also like surfactants. And these are like lots of salt, detergents. And what these do, so if you decrease the pH or you have high pH or you have high temperatures, then you can break up the tertiary structure. In tertiary structure, we've talked about that. Oxidizing agents as well. They will break up your disulfide linkage. And so what you do is you disrupt that, you lose that structure. You lose the structure, you lose the function, because now it's not folding properly, and this is disrupting the R groups, those amino acid side chains. Think about a hard-boiled egg, okay? So you have the egg when it's nice and soft, and then you hard-boil it, okay? This is denaturation, too much high temperature. The egg is protein, and it has a different consistency if you crack it open versus when it's hard-boiled. And that's because it's been denatured. Those tertiary structures have been disrupted. This is, most of our body operates with enzymes. Proteins, enzymes are made up proteins. And these proteins have a certain fold and a certain structure, and if you do denaturation, different temperatures, like your body gets a temperature, then the problem is it's just like cooking your egg. You destroy your functioning of your proteins. Okay, read your chapter, and we'll see you tomorrow.